Professor Zhigang Yao, first speaker in the afternoon session. He received his PhD in statistics from University of Pittsburgh in 2011. After that, he moved to one of the famous universities in the world, the Swiss Federal Institute of Technology. And then he worked with our president, Professor Zhigang Yao, on manifold fitting and research that interface between statistics and geometry. And he currently affiliated to the National University of Singapore. Please warmly welcome Professor Zhigang Yao to make the talk.
All right. Thank you very much for the opportunities. It's my great pleasure to be here. I'm going to talk about the things that I started doing since 2014. This will be a high-level talk and trying to cover some of the topics that I've been doing on the manifold fitting. I am a statistician and trying to study things, developing methods using geometries, because I think that many of the, I would say most of the statistical methods are very centering around using subspaces to approximate things. Most of the time, I would say 90% of the time, these subspaces are linear subspaces. If you look at the very high dimensional data, linear subspaces are useful, but when curvatures came in, then you probably wanted to looking at the curvatures as well. So in other sense, developing non-linear methods are actually of great interest for me. So I just wanted to start from a very simple example. I assume everybody here knows, if I give you a n by p matrix, and p stands for the number of the variables and n is samples, and you can always do single variable decompositions, or you can just do principal component analysis or SVDs. No matter how you do this, eventually you're gonna get a sequence. For example, if you use I would say the principal. If you simply use SVD or principal component analysis, I would say, you basically get on all these eigenvectors and order them using the eigenvalues, and these D1 or D0 up to DPs are a sequence of these eigenvectors. In other words, specifically, these are the subspaces spanned by the first K. I would say, these eigenvectors. If you look at the principal component sort of analysis in a backward way, for example, we can get I would say, if there are p eigenvectors, right, then you can get p minus 1 eigenvectors, p minus 2 eigenvectors. Then eventually every di here stands for these linear subspaces spanned by the first k eigenvectors. If you look, as I said, if you look at these things in the backward way, so they are actually nested. But unfortunately, also fortunately, these are all linear subspaces. As I said, most of the time, if you really wanted to explore things, sometimes the linear subspaces are not actually enough. So we were actually looking at a non-linear subspaces. Actually, we are actually doing something called a principal nested submanifolds, where each of these subspaces are actually manifolds. This means that we should be able to fit a lower dimensional manifolds out of any kind of dimensional data. So I will explain what do I mean by manifold fitting for fitting each of the dimensional manifolds. So this is the motivations. Okay, so in other words, PCA can give you a sequence, again, a sequence of these nested linear subspaces. So we will be able to get a sequence of these non-linear submanifolds. And I'm actually going to talk about how do we actually construct these non-linear manifolds. Eventually, try to improve, I would say, developing a new framework of how do we actually do decompositions for covariance matrix. And this eventually going to be, I'm actually going to talk about this one or two applications that will be using the manifold fitting on single cell, or also this metabolism data, I would say the data sets. So most of the time I would say that, okay, most of the people, when you try to do, I would say GAN, or, generating some of these important images, and these are the OpenAI, the framework. And the data are very large in terms of dimensions, in terms of number of the sample sizes. And if you look at all these deep learning I would say methodologies, most of the time, and when you have a very high dimensional input, what people do using deep learning is trying to actually map all these high dimensional data into a lower dimensional subspaces, or when people say the lower dimensional latent spaces, or actually even lower dimensional manifolds. Depends on how you actually parameterize or how you actually do the mappings. Then you would do all these things in this lower dimensional space. And then eventually you're going to do this decoder thing and try to visualize your outlook. And then, as I said, people have different names for this lower dimensional latent spaces. And is it even a manifold? Can you actually prove it's a manifold? This is a very different questions. So what we'll be doing is not just project the data into a lower dimensional latent space. We'll try to do a fitting of the manifolds behind the high dimensional data, where the manifold actually is still living in high dimensions, but with much, much lower dimensions. So we're actually we will be able to prove that the lower dimensional latent space is indeed a manifold and try to explore that structures. So this is hopefully trying to improve some of the methods from this AI areas. They still have some shortcomings. For example, when you have this kind of videos, we will see that for example, if you train the neural networks and for example using CycleGAN and so on, then this is the image coming from the Sora. Then you will see that all there's actually a leaking of the red lines, which means that in the lower dimensional embeddings, there's still some room to be improved. So along the lines, I just wanted to mention that there have been some applications ever since 2019. I had my first manifold fitting papers from 2019, but that paper took us honestly 4 or 5 years. Then after that, we have been implementing this manifold fitting technologies or techniques to applications such as machine learning CycleGANs and also single cells. Also the one I'm actually going to talk about called basically it's just an implementation of the fittings on this metabolic manifold theory. This is not a mathematical concept. It's just a manifold fitting onto this applications. This has been, some of the work has been a joint work with STL and also my students, postdocs, and from National University of Singapore. So motivations. So why geometry matters? So this is an extremely simple case. We've got a Swiss row here. There's a kind of data. So different color stands for different clusters, right? And then this is a R3 space, and there's a Swiss row there. So it's going to be roughly, a two-dimensional manifold. And if you wanted to do clustering, suppose we don't know the labels, you wanted to do clusterings. Right? So what people do is, okay, we're just going to destroy the Swiss row, make it flat. Basically, you will flatten the whole Swiss row into a two-dimensional piece of paper using some loss functions. Hopefully, you can preserve some distances. Okay, this is called dimension reduction. This is how canonically people will be doing. Flatten the whole Swiss row, and then you will apply all these clustering methods on the reduced dimensions. This is okay, but we believe that if you destroy the Swiss row, then you destroy the geometries. So what we'll be doing is that using all these points, we're trying to fit a two-dimensional Swiss row recovering from the data sets. Then we're just going to do the clustering directly on these points lying very close to the Swiss rows. So recovering the Swiss row from the data sets is the process we call manifold fitting from the high level sort of point of views. And then we're trying to actually fitting the data, again, fitting the manifolds exactly in R3, just putting them down into a two-dimensional piece of papers. Okay, so this is the line of thought that I just wanted to make sure that everybody here stand on the same rows. We do not do the dimension reduction literally. Instead, we're just going to fit a Swiss row in the R3. Hopefully, the Swiss row will actually fit. We can prove it's a manifold. And then we do everything based on the Swiss row. Okay? All right, so there are some different kinds of data. For example, if you're looking at, I would say the earthquake data, where basically if you model the whole Earth as a S2, a ping pong ball, basically, and then all these the coordinates of this of this earthquake X, Y, Z has to somehow obey that. They're lying on the S2. Basically, the sum of the x squared plus y squared z squared has to be equal to the square of the radius of the Earth. This is basically what it describes as a known manifolds. We know points on the Swiss row. Sorry, we know points on the S2 and trying to fit in something on these points. For example, for this particular case, for the earthquake data, the people wanted to fit a curve, following the main channel of this black dots, and the curve has to be exactly on this S2 here, the ball here. Okay, the curve is not going to go above or beneath the Earth. Not going to get into, deviate this whole the S2. The second case is that when we have M by P matrix or P by P matrix in general, right? You can always making them as a vector, but because of the multivariate data on the nonlinear constraints, that we can actually see every single M by P matrix as a point on a lower dimensional manifold. For example, this is a special case. As again, I'm going to talk about covariance matrix. When people talk about covariance matrix, there's many people in data science or even in statistics, they are studying from the covariance matrix. Covariance matrix is symmetric. So, for example, this is a 3x3 covariance matrix. If you work with tensor data, this is just one example. It's a simplest case, 3x3. So, because of the symmetry of this matrix. So if I let the P3 be a space of the all PSD defined by 3x3 matrices, then even if you line them up, making it as a 9-dimensional vectors, and the P3 is actually basically a subset of R6 because of the symmetries. But P3 is still too big. Why? Because it's actually even if it's actually convex, but it's actually not linear, because if I have two covariance matrices, D1 or 2, then if you do combinations, this guy is not going to be in the P3s. So which means that if you completely develop your methods using a R6 dimensional Euclidean methods, you still have some room to improve. So what I will view this problem is that if I can develop something, for example, PCA, that can be implemented on a manifolds, actually we can prove that every covariance matrix of this type lying on a cone, that cone living in R4. So if I have a way to fit in something or doing a canonical PCA, which didn't exist on the cones, then this can give me some advantage comparing with the usual PCA, which basically decompose the R6 using the first 1 or 2 or 3, 4, 5, up until 6 eigenvectors to do compositions. All right, so this is the abstract of doing the so-called clusterings in high dimensions. The two kinds of data, different colors stands for two different clusters. If I wanted to do separations, meaning if I wanted to do a unsupervised learning, I would say the clusterings, what people would do basically in deep learning is basically transform every single point here. This is a very high dimensions. They also transform them using some functions, using different layers to some other spaces determined by these layers, all right? And try to do business over that space. The come back to here is still going to be curved boundaries. But for me, we will actually do things. We'll fit the manifold behind high dimension first. And the manifold is actually with dimension very low. Then we will do the boundaries directly on the fit manifolds. So in other words, we're fitting the manifold in ambient space, composed by all the features of these variables. And then we'll do clusterings directly on the manifolds. And we call this thing as actually something called the principal boundary and so on. Eventually, if the manifold actually collapses into a Euclidean space, we can still prove that this kind of boundaries will actually coincide with the PCAs. So on. All right, so this is some sort of I would say the warming up before I officially talk about the manifold fittings. So 2016, when I was looking at, I actually did a very extensive search on the manifold learnings. I've actually noticed that most of the papers talking about manifold learning from the late 90s to 2010, almost 20 years, they basically said that, okay, if there is a lower dimensional manifold, but if you have a high dimensional input, let's say RP here, P is very big, if there's a lower dimensional manifold with very low dimensions, all these manifold learning approaches will actually output a lower dimensional vectors by matching some distances. So this is, to me, is just a embeddings. You do not actually necessarily estimate the manifolds. So to my surprise, that most of the methods didn't actually estimate the manifold. In other words, they didn't actually give you an estimator of the manifold lying behind the RPs. The only output is RP prime here, where the P prime is much smaller than the P's. Hopefully the P prime is very close to the small d here. The d is the dimension of the manifolds. They do not actually give the estimators. The actual manifold fitting, kind of ideas, the papers starting from 2012, and the two papers, very abstract with very restricted assumptions, talking about how do you actually recover. They actually call this as the manifold estimations officially. And then Pfefferman, who was a field medalist, also with his postdocs, trying to relook at the problems around 2016, and they come up with some ideas how do you can actually fit the manifolds behind a high dimension. They also prove that it's indeed a manifold. It's not just a lower dimensional output. And then I had a paper starting from 2019 with my postdocs trying to improve their papers, and after that, then we have been doing all these things and trying to even improve the bonds in terms of the distance bonds, and with all these applications and so on. So I would just kind of very highlight why this matters in terms of improving the analysis tools, because most people doing dimensional reductions, they rely on a very specific loss functions. And as I said, doing the dimension reductions really depends on how you actually view the object functions. So what we want to do is actually doing this dimension reduction using a geometry in the ambient space. So when I actually doing my sabbatical 2022 at Harvard, someone from medical school wrote to me, maybe you should do something very applied that everybody can use. So this is a single cell data sets. These are all real data coming from, I would say, all these big papers, Nature, I don't know, the Nature Cell Science. They're actually looking at this single cell RNA data. So we basically collect all these things. So number of the variable here, okay, is which I call features here, is 15K to 40K. Is actually pretty big. So these are all the gene expressions. For example, the first data sets here. So the number of cells here are actually 3. And the number of, so the number of cells actually is 704, and the number of the gene is 38,000. So this number means how high the dimension is. This number means the n here, okay? And these are the number of cells determined by the technicians, which we're not gonna actually use it. So the goal is that if I can fit a manifold on each of the data sets, be careful, the dimension is huge. Okay, we're not dealing with R4, R6, we're dealing with R30K, okay? And most people would do, okay, we're just gonna cut down the genes, selecting the most important genes. No, we're not gonna do it. We're going to fit a manifold where each of the points existing in the 38K dimensions. After the fittings, every point will be pushed to the manifold as close as possible, and that manifold living in that 38K dimensional spaces. Okay, so if I do that, if I had a way to do that, then I will actually do the unsupervised learnings, and then I can improve the errors. If I have time, I will actually show you the results. So basically, if you view every single cells in very high dimension as a point, then what we do is trying to push all these points in that existing ambient space, okay, to the underlying manifold as close as possible. Again, we're not going to cut down the dimensions, okay? But the manifold that we actually got can be proved as a manifold with also a very lower dimensions. So this is a fundamental difference between the fitting with the so-called manifold learnings, which basically will reduce the dimension from the P to P primes. So most people, like I was not an expert in a single cell, but I actually look at some of the very successful pipelines. They were basically cutting down the genes, for example, from 38,000 to, I don't know, 100. And naming all these 100 genes are very important, and they do business after that. No, we're not going to do that. We're going to do just using the M by P matrix. I'm going to return you the same M by P matrix. And I will claim that on the returning M by P matrix, every single row there as a point will lie much closer to the underlying manifold. And I do not need to determine the dimension of that manifold. Okay, that's the general picture of the manifold fitting. Okay. So recently, then some of my postdoc and students, they were very interested in this so-called UK Biobank dataset, which actually I had a chance to actually pay some money to buy it, by the usage of that. So they follow almost like 500,000 people. Okay, it's very high quality, I would say, level of the data sets. They follow them. They know how they sleep, how long they sleep. They know everything about them. Like for example, the gene data, MR data. They follow them for 20 years and so on. Then they release the results so we can buy it. So it's the data set I'm going to talk about after this. So the manifold fitting is that we got this data for 200,000 people. It's a huge population. So it's a 200,000 people, and we follow their blood sample. People say, what's the big deal of the blood samples? If you go to the hospital, you get your blood tested. The thing is that they managed to, using MRI, to scan the blood samples and return you the 251 biomarkers. It's very important. This basically tells you there are actually 251 biomarkers in the blood. And then we're going to get a matrix with this N and this P here. But we're going to fit a manifold behind this 200,000 multiplied 251 dimensional matrix. Then I will try to explain why this matters in terms of doing this classifications. All right. So some notions. So for mathematicians, if you have for example, this 2016 paper that I mentioned, the just performance. If you're given a metric space and you know that the underlying dimension is small d, which is much smaller than the ambient space capital D, then to them they want to get a manifold estimator. Also they also will prove that it's indeed a manifold, and also control the dimension of the M hat with small d's. In my 2019 paper, I actually wrote that. I actually basically translated the problem into a problem which we can much more better in terms of the understandings. So there's a manifold behind the high dimension data. The dimensional ambient space is capital D's. So the axis, which has no noise on it, they actually line on the manifold with a metric, with a distribution basically. But most of the time we will observe noises. So what you observe is a Y here. So we want actually to construct a manifold estimator and then try to bond the M hat with the M here. Be careful, we're going to give a manifold estimator, not just output in lower dimensional output. That's very different. Okay, and if we control the distance between these two guys, then you can write a few papers. All right, so to highlight, and this is the manifold structure here. So these are all high dimensional data points. So all these isomap and, for example, local embeddings, they just look at the neighborhood, try to use PC or whatever loss functions, try to map all these points into a lower dimensional P prime dimensional subspaces. Once you are here getting all these red dots, you can't go back. So this, to me, is embedding approach. And then what we're trying to do is that we're still living in the high dimensional ambient space and projecting all these points, hopefully, to reaching to these red dots. And we can, as if we can guarantee that all these points can lie very close to the underlying manifold in the original ambient space. then this will be very good to us. Then we can even fit a smooth manifold based on these red dots. So why we want to do that? I mean, if we can actually get a smooth manifold, we can actually do interpolations, and everything here on this part C here, living in the capital D ambient space. So we can do whatever you wanted to do. We don't even need a decoders to visualize your results. No, because everything is already in high dimensions. Okay. This is the very earlier version of the model setup. So we've got a point here with eyes, and there has two components. One is xi, the second is Ci here. So the important thing about the Ci is that Ci has densities, for example, following some kind of distributions. So we observe the yi. The goal is trying to using the yi's and to recover some sort of estimators. The M is not flat, so the reach of the manifold measures how flat the M is. So we had to give this M a lower bound. So you can also assume it's a C2 or C dimensional manifold with a small d here. When people, trying to keep asking me, can you actually make it complicated? Yes, you can do it. But this is just one very early version of the setup. Okay. So this 2012 paper that by Larry Bossman and also the Christian University, there are two authors, actually four authors. They assume a very, to me, it's very ideal assumptions. When I say ideal, I mean it's actually very restricted. They assume that the Ci, the noise, is orthogonal to the tangent space of the M for every single xi here. The xi is noiseless data points lying on the M here. So the most important thing is they actually assume that Ci is bounded. If the Ci is bounded, basically you are looking at data points roughly at a tube of the manifold. So this is the manifold. Remember the picture I actually gave you. So if the noise is very bounded, then roughly you are looking at a tube surrounding the true manifold. Then of course you can come up with some estimators. This is too restricted to me. So it's actually better to work with Gaussian noise, which is unbounded. But they come up with this, I would say the minimax bound, which means that the manifold and also the underlying manifold, if the convergence, the minimax rate is already 1 over log n when the n is sample size. So you need a huge number of n to recover the things. So which was not possible for them. So at that time, I would say around 2012, Larry declared that they're not going to really work on the manifold estimations. They care about something else. For example, the reach estimations. They will just sacrifice some bias here, so actually to make it much more convergent. But to me, that these are just one of the arbitrary estimators. What I actually care is the manifold estimators, not just the ridge estimate. The ridge is not a manifold. So before I talk about my approach to deal with the manifold fittings, I just want to highlight the key idea of the Pfefferman 2016 paper. For example, this is the underlying manifold, and you want to actually to push the y here. Y can be any point, can be your sample point, or any point you want to push the manifold, but this is called manifold fittings. The yi, yj's in this picture stands for some data points, some sample points. So what they do is that, okay, they just look around here with a radius r. Why the r is square root of the sigma. The sigma is the noise levels. So when you have the sigma noise levels, you look at the radius at the square root of the sigma, so that when you compute the covariance matrix within this local neighborhood, you can get some eigengap. That's all. Okay, and then they also need this number of the size of samples, depending on the intrinsic dimension of all of the manifold small d, so that they can do this business. So what they do is that, okay, if we've got these 2 points here within this neighborhood, center on the y here, so you can decide using these local neighborhoods to actually project the y using this directions, using this directions. And then you can shooting down the y onto this y star, getting this so-called black arrows. And black is just, this arrow is just with the average of all these different colors of the arrows. Of course, the Fy is just a weighted average of these vectors. And this one, the black one is not really shooting to the Y star because we don't know the Y stars. So you have to do one more projection to actually, make it much more realistic. So there will be 2 steps of this projections. Hopefully, we can shooting this Y onto a point which is very close to the Y star. So define the manifold estimator as that if the black arrow, this is still going to be, no matter how high the dimension is for every single data point, the black arrow is just one-dimensional vectors. So if the norm after the second sort of projections becomes 0, if the norm becomes 0 of their vectors, you're arriving at the manifold, getting a nice estimator for the Y star. If I have a lot of Y star, I can recover the manifold. So this basically is actually a level set, making sure the norm of after the last second step of the projection is 0. To prove that, okay, within, with high probabilities, for example, because we're trying to deal with random errors, of course, and this can be bounded by some kind of a square of the R's here. So the R square basically tells you it's an O sigma bond by looking at the manifold fittings. Well, there's something that we can actually sort of, improve, but let's have a look at this thing first. So this is a circle in this picture. Okay, the circle stands for the perfect manifold. So we've got this sort of data here in grains. So this is just a toy problems. If I run this manifold fittings using their approach, what you try to do is, everybody here can just spend some time and write a very simple code. So what they do is they would try to use a sequence of these balls to truncate the so-called data points. And then within each of the balls, you can use PCA to estimate the local structures using PCA or whatever you wanted to use. And then all these blue, all these light blue stands for the lower dimensional PCA results. These are the results telling you how do you actually try to project the points using the blue and also the red ones. These are telling you what is the sort of estimation direction you can actually project all these points. So if I zoom in one of the whole things, so after I take off all these points, these are all local structures, estimate from the data sets. In this case, it's just all these green dots. So the green one is the Y's. I wanted to project my Y onto somewhere very close to the circle. Using, for this particular case, if I'm standing here looking at the run here, there are actually 1, 2, 3 different linear subspaces I can borrow, which means I can have Yi, Yj, Yk. So these are the 3 lower dimensional structures. So I can forget about everything here, everything here, everything here. So I can do all these zoom ins, and these are the all weighted average of these vectors. So if I plot the norm of the last second projections, and you will see that for points around here, very far away from the circle, then the norm after their approach is still going to be very big, not possibly getting very close to the manifold. For all these points surrounding the circles, then you probably can use their sort of algorithms to getting very close to the Y stars. So why we actually wanted to improve the methods, because I think that even with the unbounded noise, such as Gaussian noise, okay, if I got these points, data points, surrounding actually in the truncated by this Euclidean balls, then the mean of these points, the samples, even with the unbounded noise, still going to be very close, because of Gaussians, very close to the manifolds. In this case, I will simply take the Fauché means. The Fauché mean is just the mean that you try to compute for points lying on the manifolds. So you're not going to compute a sample average, like you add up all these x's divided by n. No, these are called the Fauché means. Fauché mean is just an analog of the sample means on the manifold. So I claim that the Y tutor is still going to be very close to the manifolds. So why does that matter? That actually matters because you don't need it to actually use these projections. So if I got this Y star very close to somewhere here, then I can just compute the tangent space at the y star, which could be very parallel with the tangent space of the y star here, not theta here. But actually it turns out that I don't need to do that. So the B here is just a Fisher mean here. So that tangent space is very close to the B star here, which is exactly on a manifold. But actually I can just compute the normal directions. That directions were shooting down directly to the manifolds, towards the manifolds. And you can see that bias is very much smaller than the bias of the Pfefferman 2016 papers. So this is why a simple improvement could actually give us a much better, I would say, the practical results. So the manifold estimate here is that I can just taking down the y minus the y theta and do this one projections, making sure the norm is 0, then we're actually arriving at the manifolds. But to be careful, this is not going to actually improve the bonds theoretically. We're still going to get R squared, which is O sigma bond. But practically, we can still get a much better results. But again, this is the 2019 papers. I just wanted to mention a few things about this. How do we actually do better, beyond the 2019 papers? The idea is very interesting. So we're not going to use the Euclidean ball to truncate the points, because for everybody, if you look at neighborhood, we actually look at a Euclidean ball. You set the radius of the Euclidean ball and try to get all the points. The Euclidean ball is actually will be very much bounded, because the PCA will be bounded by the Euclidean ball. In order to improve the fittings from the, let's say the O sigma to a high order, yeah. We assume the manifold is known, but we assume we know nothing about the manifold. I will estimate, but I don't know how to parameterize the manifold at all. So on every high dimensional space, if you have a data cloud, if you have enough points, okay, there will be a manifold existing. And there actually can be many different manifolds with different curvatures. And that was a hypothesis. And the hypothesis recently was actually proved by Pfefferman in his paper. So you can always push the point to a underlying manifold no matter what. That result was not our results. That was theoretical results.
So in that heatgram heat map plot,
yeah,
how do you obtain this heat map? You assume the manifold is known?
No, we don't assume. I assume nothing about the circle. So this is showing you, if I take every single pixel here, how do I get back at least a version of the circle? By computing the results that we got, the norm of it. So the light color telling you that for the points here, if I follow Pfefferman's paper, that approach, the norm is much closer to the zeros. So this is how these areas are actually very light. If you got points here, you can't do a better job, because this color is actually blue. Blue stands for norm is very big after 2 steps of their projection. So the goal is to try to recover the circle, but we do not assume the circle at all. Yeah. Yeah.
Yeah, my question is whether this is a consistent. If you have a 0 error,
if I have 0 errors, then every point is presumably onto a manifold.
It's going to be moved or not moved. You're going to still project it to some linear space, right?
No. If there's no noise, then, for example, if you're shrinking the points, okay, and your observation will be roughly here, very surrounding the circles.
Does it depend on the choice of epsilon?
Choice of what?
The radius of the ball?
Yes.
Depend on the choice of the radius?
Yeah.
How do I choose epsilon?
That's a good question. I will talk about it. I will talk about it. This later. Any questions? Otherwise, I'm gonna go on. So 2022, then the world was in pandemic, so we actually thought that FFM, sort of get another draft trying to improve the bonds. So we actually did something similar. For most people, if you have a point here, okay, to do the manifold fitting, or I would say to do the recovery of all the manifold, you're shooting this point to getting this blue dots and making sure the blue dots is very close to the manifold. That will be it. Okay, that will be it. You can write many papers trying to change the way you try to project the point to getting those blue ones. But the goal is here. The blue one so far is at a Hausdorff distance, which is O sigma from the underlying manifold. That was the best bond. But if you wanted to actually do one more projections, you actually needed to change a way of actually truncate the data point. As I said, doing a local ball is bounded by these eigengaps. So we're not going to use the local balls. We're going to use a cylinders in terms of the product of the two spheres. Why cylinder? Which is interesting. So let me tell you what this thing is. So this is, the blue dots is guaranteed from my 2019 paper or any paper, for example, the Pfefferman's paper. So if I construct a cylinders, and then if I shrink the cylinders, okay, if I shrink the cylinders, making it thinner, and then you see that all these points, all these gray points that I can take average upon on, the direction of the axis of the cylinder will be very much close to the actual projection I wanted to further project the blue dots to the manifold, arriving at the green dots. And this basically, in a very high level, is actually called the local constructions. And why this actually matters, we had this results. So Fy is the blue dots, okay, and this is the underlying. Sorry, this is the point you wanted to project to the manifold. So Y star is in the population ones, something I don't understand. We don't know about it. This is target. The PCA can only do O sigma, as I said, because PCA need an eigengap. Once you have a radius, which is square root of sigmas, you are bounded by the PCA performance. So to bond these two angles, to bond these two vectors, the cylinder approach can basically guarantee that the angle between these two vectors can be actually bounded by sigma times a very small log terms. And that one, combined with the previous, I would say the blue dots, can give us the manifold estimations, which is at the level of the sigma squared times this log term. If we're dealing
How
with...
to fix the upper bound, Professor, excuse me?
Sir?
The upper bound over there, how did you fix that? C sigma square log 1 over sigma.
Then this is the whole 2022 papers. So basically, I would say that, okay, previously we've got this, actually we introduced this new projections, and we take another averages. As I said, that the key thing is that if you simply using the Euclidean ball to get this neighborhood, you can't really beat this. You can always get O sigma no matter what. And this is further project the blue dots to the green ones. If you keep iterating the blue dots, you won't be actually getting close to the manifold. So you need a one-step projections. So far we can get O sigma times this log term. If you are dealing with vanishing noise, then this is already good enough as compared to the sigma squares. Okay. All right. So, and after that, I will skip some of the details. After that, we applied this thing to different machine learnings or single cell and so on. Recently, we have a new pipeline called a single cell scope. And that cell scope basically is a new pipeline trying to compete with everybody. I was just going to talk about this so-called metabolism biomarkers very quickly. Maybe I should finish this first. So the so-called cyclogram in machine learning, they're trying to, learning mappings between the Z and Y domains. Z can be low dimensional, high dimensional, Y can be high dimensional. Well, the cyclogram trained the two generators, okay? And so 2023, I had a conference at Harvard, and actually someone very distinguished professor in statistics told me that maybe you should combine this manifold fitting with the cyclogram, trying to help them to do the convergence. So I thought about it. This Y is in high dimensions. So if you're talking about, for example, images, these are all long, very high dimensional vectors in high dimensions. The Z can be very low dimension Gaussian noise, and you can actually run the mappings. The generators basically, after training, help you to generate these images, which will look probably very authentic. And you actually train the whole network. So we actually adding up a manifold fitting step to this Y dimension, to this high dimensional Y. Why is that? Because the Y is in high dimensions. The Y tutor is getting, if I have actually a generators, we can actually generate the Y from a fake Z, and then we can actually come back to regenerate these fake data. But for these data, no matter it's a fake or not, it's always going to be in high dimensions. So as long as they're in high dimensions, we can fit a lower, we can push all these points within their domains onto a very close manifold. And before we pass this results to the discriminators, and this is simply a combination of the cyclogram with the manifold fittings. And then you can actually do many things. Well, we're just going to skip most of our sort of material and talk about this toy problem estimations. So these are the L data. You rotate the L's, putting some noise on it. The manifold fitting not only can give you the denoised version of the L's, which looks not too bad, but also we can do, because we actually recover the manifold, so we can do interpolations. So if I fix these two images after the fittings, we can actually do the interpolations. All these 1, 2, 3, 4, 5, 6, totally actually the 12 images between these two folks are recovered from the fit manifold. And these images were not in one of these original data sets. Okay? And the single cell, I'm just going to very quickly mention the first error, so the first data sets. Again, it's a 704 sample, and the dimension is 30,000. Okay, it's huge. And the technician told me, okay, there are actually 3 different clusters. I don't believe it. But this is just showing you, okay, there are actually 3 different clusters according to them. They spend a lot of money is actually working on this thing and trying to, manually determine which cell belong to which. They're totally different, 3 different cell types. So what we do is that, for example, this picture, okay? The cells in high dimension, 38,000, so we can use a low dimensional visualizations like everyone else. For example, they use t-SNEs to get 2-dimensional representations so you can view it. And because we know there are actually 3 different cells according to the technicians, we can color them. So you will see that in this lower dimensional visualization, there are actually some sort of, I would say they're perfectly mixed together. Then when people can use deep learnings or whatever to tune them and eventually separating these things in the high dimensions. So what we did is that we just fit the manifold on the data with 704 multiply 30,000, returning back a 704 multiply 30,000, and I will just use t-SNE to revisualize it. Again, just visualize it. We didn't do anything. Okay, we didn't do clustering or anything. This is the result for the first data set that I just mentioned out of 25 data sets. After the manifold fitting, for the 704 multiply 30,000, the lower dimension t-SNE visualization looks like this. Of course, we know the labels, so we're just showing you this. So you will see that for the fit manifold matrix, these data are actually clustered together. Again, this is not a classification. results. This is just a result showing you what I got. You still have to apply whatever k-means or anything to classify. There's little difference here. You will see that even so, these points, according to technicians, they belong to same clusters. There's still some sort of gap between these two guys, indicating that there might be some chance we can actually figure out some very rare cell types. Again, this was 2023 January results. I thought that was just a coincidence. So we ended up coding everything on the 25 data sets, single cell with dimensions from 15K to 45K. This guy, this row stands for all these deep learning approaches, for example, using autoencoders, because they have to ask us to compare our approach with the pipelines. And this one, the second column, is that if you fit the data using the manifold fittings and how much you can improve. So basically, out of the 25 data sets, we can improve upon existing platforms by at least 25%. Not in terms of the misclassification error, in terms of the so-called ARR index. If we simply rely on the arrows, then we can actually improve beyond 35%. So out of the 25 data sets, we get this performance. Basically, it means that there must be something meaningful we have done. Otherwise, we can't actually beat this autoencoded stuff. Now, the last thing I wanted to mention is the so-called UK Biobank metabolism data sets. 200,000 people, we follow them. The 251 biomarkers from the blood. We separate the 251 variable, the features, into 7 different groups, giving us C1, C2, C3, C4, C7, whatever. Which means that the number of the features here plus the number of features here plus and so on will give us 251. Okay, so we apply, again, it's very simple. We apply the manifold fitting on 200,000 people, multiply this number of features on here and here. We actually do this 7 times. Very simple, just 7 times fitting on 7 different matrices. And we got 7 different results, which I call M1, M2, M3, M4, M5, M6, 7. Again, we do not reduce any features. So the M1 had the same size as C1, M2 had the same size as C2, and so on. It's just a denoised version. You can simply see that in that way. People say, so what? I mean, we can also do that, right? So the 200,000 people, we subsampling. Actually, we will fit the whole thing on the 200,000 people. So we subsampling, let's say 50,000, and visualize it. If I take 200,000, then you will get much dense pictures. So the number of points on this picture tell us the results for, I would say, the 50K people. And this was the C1, or this is C1s. Okay? If you use autoencoders, you use different, whatever fancy approach. If you wanted to visualize them for your result, you get this. The number of points here, still 50K. We fit the C1 using the fitting, get the M1, and visualize it using the UMAP. Sorry, not using UMAP. On the M1 here, this is what I got. Very different from if you use autoencoders or whatever approach. And people will say, so what? I mean, you actually separate them into 2, there are 2 different clusters. People will say, okay, so what? I mean, there are 2 clusters. Your method classify, or at least when you see them, you will say, oh, actually, there are 2 clusters. Be careful. We use this metabolism variables, but we also had a huge number of the history of the medical history of every person in this picture. So the grouping effect we actually got from this manifold fittings, we cross-check every single people here, every single people in the second classes. It actually turns out for the first 50 most interesting disease. Be careful, we got everybody who actually gets sick, who actually got whatever cancer, high blood pressures in the entire data sets. We cross-check all these people. The risk of getting the first 50 diseases is much higher, significantly higher than these people. There are actually 4,000 people here. There are 46,000 people on the second clusters. So which means that the thing we did using the metabolism data sets can give us a guidelines to cluster people without knowing their medical histories, which is very interesting to me because this is telling us that this can be served as a way to predetermine obviously the chances of these people who get whatever diseases. So actually, the data set has 1,000 disease the indicator for every person following the 20 years. And this is just a result. And then we got, out of the, actually, I would say the 7 different groups. Okay. I don't know if I have this picture here or not, but maybe it's not here. Anyway.
You have 5 minutes, Prof.
Okay, yeah. I'm actually wrapping up very, very quickly, very soon. I tried to find this slide, but I can't find it anyways. Sorry. Anyway, so the result I just mentioned here was actually coming from the M1. For M2 and M5, we also discovered that patterns. We found similar results on the M3, M4, M7. But for the paper, we only summarize the performance of M1, M2, M5, which is already enough to publish a paper in the scientific journals. So my conclusion is that there definitely has been something we have done correctly, and then by looking at how fittings for the high-dimensional data will be still a ongoing research. And the thing I just mentioned here, if I can fit every single lower dimensional manifolds, how do I actually get a new framework, making sure these are all nested within each other? And this will give a very complete, fresh way to actually replace the PCAs. Okay, and then this probably just gonna be my main theme of the talk. And thank you very much for the attention.
Wow, it's awesome. So questions?
Thank you. My question is, when you measure the distance from your point to the manifold, you always consider Euclidean one?
Hausdorff distance.
What?
Hausdorff distance.
Hausdorff.
Yeah, because we're dealing with 2 sets. So it's a Hausdorff distance.
And did you try the method of width theory and results like Johnson-Lindenstrauss lemma?
I haven't.
Never heard it.
No.
Okay.
But I would definitely would be very interesting.
So after you fit your model, do you know the intrinsic dimension of the manifold you fitted?
We don't know, but we can estimate. So most of the approach, they need an input.
For the single cell data.
For the single cell, here's my response. So it's, for example, the first data set is 38,000. So if you look at the results that we actually fit, locally, if you compute the graph, the rank of that graph will not be exceeding 15th. So which means that usually the dimension of the manifold is not going to be exceeding 15th, even for the very high dimensional input. And my 2019 paper, we need input parameters for the fittings, but the 2022 that I actually show you right now, we don't need to know the intrinsic dimension of the manifold.
That's fair. So the for the single cell data specifically,
Yeah.
people know that some genes are more trustworthy than others. For
Yes.
example, many genes have pseudogenes and their expression measurement maybe have large variance. So when you fit the manifold, is there a way for you to take that into account?
Yes, we do. I mean, actually, for the thing I haven't discussed, the cell scope. Actually, the manuscript is already online. So in single cell, people are very keen in describing gene as I would say the housekeeping genes, and some of the genes are very important and so on. But all these are from some certain sort of variable selection methods. So it's better for us to actually have a tree structure to zoom in. But right now, the cell scope, if you look at our pipelines, then we can actually determine all these important genes at every single step when you zoom in. But I decide not to pay too much attention on selecting the most important genes. Because selecting the most important genes with respect to this, I would say it's just like a variable selections. And then sometimes can be subjective.
Okay, a related question is that for the UK Biobank data,
Yeah.
and the samples are heterogeneous, and which means that some sample might have fall in different ethnic groups, so they are not exchangeable. What do you do with that?
Yeah, that's a good question. So mathematically, I have to admit that everything that I actually talk about today, we're fitting a manifold. We're fitting a manifold and get some theoretical results guaranteed. So actually for either heterogeneous or whatever samples, one should not fit just only one manifold. So on a very high level, we should actually fit maybe multiple, I would say a collection of the manifold for different sections of the data. But mathematically, right now, it's still our ongoing work. I can't just say, okay, fitting one single manifold is enough. Yeah. But dealing with the curvatures of getting multiple manifolds is still in my main theory directions.
Okay, any other question? Okay, Professor, maybe is there any progress on your work with disease associations and manifold feeding? Because
Progress of what?
like disease associations and manifold feeding. While talking about manifold feeding.
Yes, actually no. Let me just go back to this timelines. I have some group members who were very interested in doing the so-called causal inference or whatever disease the inferences. Previously, this 251 metabolism data set already gave us a guideline, but this guideline is very rough. Okay, and now people are very interested. Actually, I actually got some requests asking me, can you learn mappings from these things, the metabolism to the diseases? This just my previous result just said, okay, this can be give us a rough way to cluster people, and we cross-check the risk. But actually, it's much easier. I would not so much meaningful actually to finding a mapping between the metabolism with this disease type and getting the associations between the disease with this metabolism. But this actually is what's happening in your blood, basically, and why or how this is connected with the disease type, which people might develop within, I would say, the next 5 to 10 years. But fortunately, I mean, the UK Biobank has everything. Yeah.
Any other question? All right.
This might be an open-ended. So, for example, I have not exactly from a manifold. For example, it's just a star-shaped thing. What if I give you this kind of data? Can you? What kind of? What do you? What can you say about it? Yes.
So So my, some people, they work on, actually many years ago, there's still some people at the Carnegie Mellon, they work on the cosmology data sets. So they got all these trees, they take all these things from the universe, whatever, all these data points. So these are the boundaries. They actually call the ridge. They need to develop some way to identify these ridge from this cosmology data sets. So your triangle thing is just one of the structure here. So my fitting can apply to any matrix, n by p matrix. And of course, for the tree structure, I can't guarantee that the thing I actually output is a manifold, which is not manifold. It's just some kind of stratification of some structures. So yes, if you give me any data points, for example, triangles, I can get you the triangle. But this is not a manifold. I can still output things. Yeah, just like this. If you give me a bunch of two-dimensional points messing up all on this two-dimensional pictures, I can give you all these boundaries.
Okay, great. Let's thank Professor Zhigang Yao once again, please.